Hello, College Biology. This is going to be a video over cell membrane, specifically active transport. And active transport is divided into sort of three categories. They all involve proteins, and active means it needs energy. So we have to have ATP playing some role in this, because this will not happen on its own. So the proteins involved are divided into three categories. We have a symporter, an antiporter, and a uniporter. So in this case, uni means one, which means we are transporting one thing. And usually, when you are transporting during active transport, you are transporting against the concentration gradient. So you have a high concentration of maybe glucose within the cell, and you are trying to get more glucose within the cell. This is not like diffusion. This is against the concentration gradient, not with it. So whatever this one thing is, it's going opposite the direction of the concentration gradient. Another category is the symporter, sim meaning with or together. In this case, we are taking two things in the same direction across with the help of the protein. Now, this, of course, will require ATP because this is active transport, but one of these things is going with its concentration gradient, and the other thing is going against it. So for one of these, they're going from a high concentration to a low concentration, and from the other one, they're going from a low concentration to a high concentration. Antiport is our third category, anti, against, so they are moving against each other. But even though they're moving in opposite directions, what's true for symport is still true for antiport. One of these is moving with their concentration gradient from a high concentration to a low concentration, and the other is moving against it from a low concentration to a high concentration. So let's look at an example of that. This is actually an example of two of the situations. First, we have the uniporter, the proton pump, that has hydrogen on the inside. We're moving it from hydrogen a hydrogen ion from the inside of the cell to the outside of the cell where there is already a much higher concentration of hydrogen. This does not happen on its own, so we need ATP to help this move along. What will happen is the hydrogen will bind with the uniporter on its own through polar dynamics, but that's not enough to change the shape of the protein. So ATP is then needed to change that shape, which then releases the hydrogen to the outside of the cell. So that's how we get the hydrogen from the outside. We need ATP to do that. Once it's on the outside, we have a symporter here, which uses that hydrogen. It uses the concentration gradient. We've got a lot of hydrogen on this side, so the hydrogen wants to get back in. So the hydrogen will bind with this protein, and the sucrose will bind with this protein, and the hydrogen in the sucrose then will have enough to get that sucrose in. So this is a co-transporter, this is a symporter, and this one needed energy. This one actually didn't need energy. This is a passive transport. It's an example of symport, but it's passive transport. But it can only exist because this active uniporter was pumping hydrogen to the outside. So let's go to our last example. This is the SodiumPotassium pump. You actually saw a video of this when we were taking our early tour of the cell at the beginning of this unit. It is an antiporter. So what happens is we have the extracellular fluid and we have the cytoplasm. So this is all inside the cell. This is outside the cell. And you'll note that we have a low concentration of sodium inside the cell and a high concentration of sodium outside the cell. So we will be moving against the concentration gradient. The other thing we're going to be moving with this antiporter is potassium, and you'll note that potassium has a low concentration outside of the cell and a high concentration inside of the cell, and we're moving it from outside to in. So in both cases, this time, we are going against the concentration gradient, so we are going to need energy for this. So the first thing that happens is that the sodium ions are attracted to the protein due to its polarity, but that is not enough to change the shape of the protein and release them to the outside. So we've got to get ATP involved. It's got to leave phosphorus and then split into ADP because now it only has two phosphates. But that will cause a conformational change for the protein. It will change its shape. When it changes its shape, its polarity dynamics changes. It releases the sodium. So now we've dumped more sodium to outside of the cell. Protein's just sitting there, but it will attract potassium. When it attracts potassium, it will renature to its original, well, not quite, but it'll change its shape, it will release, and then renature. So this ATP is needed for the conformational change to get us out here, and then potassium brings us back to the reset point. This is an antiporter because the potassium is going one way and the sodium is going the other way. So active transport, it's diffusion, usually against a concentration gradient. It requires ATP for the proteins to change shape properly or reset, and it involves uniporters, moving things in one direction, symporters, moving two things in the same direction, and antiporters, moving two things in opposite directions. I hope this helps. Struggle well.